Hi everyone, I'm Emma Bodley, the chair of BCARM, so the BGANZ Collections and Records Management Group, and I just want to welcome you all along to the March Plant Forum. Just some general housekeeping. If you're not speaking, please keep yourself on mute, and we will be recording the session. Thanks, Rebecca. So if you don't want to be seen on that, feel free to turn your camera off. And without further ado, I'm going to hand over to Amelia to start us off.
Thanks, Emma. My name's Amelia Martin-Jensen. I'm the project manager for the Germplasm Guidelines revision with the Australian Network for Plant Conservation. I'd like to acknowledge the traditional owners of the land where I am today, the Darug people, and pay my respects to all First Nations people, particularly those involved in conservation in Australia and New Zealand. Thanks for joining us for our second joint BCARM Plant Forum and ANPC webinar, Plant Treasures in Conversation. Today, we're really lucky to be joined by three experts in the field of plant disease and collection management to talk about biosecurity considerations in conservation collections. We have the three Bs with us presenting today. Brett Summerall will open the discussion with an overview of basic plant pathology and then talk about Phytophthora in Australia and its impacts. Bob Makinson will talk us through the precautions required to avoid introducing disease at each step in the process of acquiring, maintaining and using plants in a range of different settings. And then Beck Stanley will conclude the presentations with a case study on managing ex situ collections in the garden and nursery that are highly susceptible to pathogens and how these collections can be safely returned to the wild. Please add your questions in the chat throughout our webinar today, or raise your hand at the end of each talk to ask a question of the speakers. Emma, if you want to change slides now. We've also compiled a list of resources for you to use and share throughout the session. And remember, the germplasm guidelines are available for free download and can now be ordered as hard copies for the ANPC website. This includes guidance on protecting plants from disease and weeds, case studies on myrtle rust and Phytophthora susceptible species, as well as advice on biosecurity for various ex situ collection types. And we'll begin with Brett's presentation. Thanks so much.
Right, I shall now try to find where I put this bloody thing. No, that's not really very helpful at all. Coming up there. There we go. And I'll just do the slideshow and the beginning. And there we go. Hopefully you can all see that okay.
Yep.
No complaints. So I've got a quite a dense presentation, and I'll try to go through some basic introduction to the to plant diseases and the sorts of things that we talk about. But most of my discussion and presentation will focus on Phytophthora as a significant disease issue, particularly because of the problems that it presents to us in terms of ex situ collections, but also in terms of restoration and translocation programs in the when we're returning these plant species back to the wild. So just a little bit about plant diseases in botanic gardens. This is just a picture of our facility at the RBG Sydney. I thought it's interesting to point to just talk about the total number of species that we're working with in a typical botanic garden setup. So we're talking about 17,000 odd species, some which are extinct in the wild, a number that are critically endangered, and working across three different unique environments. So the old disease triangle, the pathogen, environment and host is really critical in that case. And I always make the point that human doctors have it easy compared to the sorts of work that we have to do. And in terms of the general plant disease problems that we're likely to see, they can range from a range of diseases that you might see on the foliage, such as powdery mildew, black spot, all of those conventional diseases that we'll see very common in a botanic garden, and of course new things which both I'm sure Bob and Beck will talk about in more details, such as myrtle rust, a shot here that Bob took of Jambos syzygium. But the ones that tend to be more problematic are those soil-borne diseases, the diseases that where most of the activity is happening under the ground, and the symptoms that we see are often quite vague in terms of what you might see above ground. Things like Phytophthora, armillaria on the right-hand side of the screen there. For one, small period of the year, you can actually see the mushrooms and know that the disease is happening, but quite problematic in botanic gardens collections, and also have the potential to be either introduced back into the wild if the hygiene protocols are not put in place. I thought I'd also mention that because many of us are growing species that have never been cultivated in the wild before, we're often working with new problems that arise. And with some of my colleagues, we've described a whole range of pathogens and potential pathogens on many of the native species we grow, for example, at the Australian Botanic Gardens. So we can have a huge diversity of unknown plant pathogens, unknown fungi, or any other sort of pathogen, but also working in somewhat of a vacuum in terms of knowing what the impact of those diseases will be in the long run. Generally, when we're looking at the management practices for disease control in botanic gardens, we tend to think of the methodologies and the practices that we do that don't involve the use of a pesticide. So this can be looking at prevention, avoidance, quarantine, and removal of a plant if it's badly infected, and particularly awareness is a really critical component of it. It's always about trying to prevent the disease through hygiene, through quarantine, so that, because that is the cheapest, most effective and most efficient way to manage plant diseases in these sort of scenarios. So just a few slides to highlight the importance of gardens hygiene, disease management practices that all ensure and are about both avoiding disease and preventing the introduction of new pathogens into a site, if that's at all possible. This refers not only to the botanic gardens that you're working with, but also when going back out into the natural ecosystems, whether it's for field work, for reintroducing plants in translocation programs or the like. Critical, particularly in you see the shot there with Costa, particularly important with diseases which have been impacted negatively. So I'm going to spend the rest of the talk mostly focused on Phytophthora, which is the most important disease of trees and shrubs in Australia and of course in New Zealand, particularly at the moment over the last few years with Kauri dieback. It has an enormous host range, but in Australia it's particularly problematic in the Proteaceae, Pachydaceae, but it's very, very broad. It's a pathogen that's microscopic, attacks the root system and limits the ability of plants to attract water and nutrients from the soil. Generally a pathogen of higher rainfall areas. God only knows what it's doing at the moment up and down the east coast of Australia with all of that. I try to avoid the use of the word unprecedented, but it's amazingly wet in those soils, and you can just imagine the movement of these pathogens through those soils. It's a pathogen that will hang around in soil for many, many years once it's been introduced. So if you get the disease or the pathogen into your collections, into your gardens, it's a problem that you have to deal with for a long period of time. And it's, of course, a key threatening process in a number of legislations now. In Australia, and I'll focus here on this because it's perhaps a little easier to work with. There's a fabulous new paper that's just come out at the end of last year, headed up by Trina Burgess, but having a number of collections and gardens people involved with it, looking at the number of Phytophthora species in Australia. So 91 species recorded in Australia, plus a few unsubstantiated. We've documented now that there are 27 species native. Cinnamomi, the poorly named cinnamon fungus, is the most important, but there are a number of other species that are important pathogens and can cause problems in a range of different species, some of which we don't know. They tend to be the exotic pathogens, in some cases been here for 100, 150 or even longer years. Huge host range, 130 host families, 416 host genera, exotic and natives, really important in ornamental horticulture and in production horticulture, fruit crops and the likes. So can affect trees, shrubs, herbaceous, annuals, all sorts of species, you name it, it can attack it. So Phytophthora literally means plant destroyer, so it's a very apt name. The life cycle is important, and it's just a few key points to raise here. Is that it is everything that you're looking at is microscopic. You cannot detect the presence just by observation. It produces the hyphae, produces the sporangia that you see on the right-hand side of the screen, which I'll talk a little bit more about in a moment, which release zoospores on the left hand at the bottom. These are biflagellated tiny spores that have the ability to swim through the water in the soil and infect the plant from that point of view. And they have survival mechanisms, a number of survival mechanisms, chlamydospores, very thickened walls, able to survive in the soil for many, many, many months and into years. So it's a persistent problem. These zoospores that are released swim through the water. In this case, we're talking about in Cinnamomi, produces large numbers very quickly. So it can multiply up and moves through the water, and you quite often see this sort of disease front moving down a slope. So in this case, the susceptible species is the Xanthorrhoeas, and you can see that it's working its way down the hill as the disease front moves through the environment there, and produces masses of the zoospores. This poor old plant really had no chance in terms of the number of infection points on the roots, and the mycelium germinates from those zoospores and it grows through the root, effectively destroying the soils. And basically you get a dysfunctional root system, unable to extract moisture and nutrients from the soil, the potting mix, whatever. The symptoms can be initially vague. You quite often see this sort of yellowing, and this quite often can be difficult to differentiate from drought, from nutrient deficiency and the like. I deliberately put this slide in because it's not a plant that we were able to detect Phytophthora from, so it's probably a nutrient deficiency, but the difficulty in differentiating nutrient deficiency and Phytophthora infection can be very problematic. This is sort of the more advanced symptoms that we might see in this particular case in rhododendrons, but in very susceptible species like Xanthorrhoeas, the disease can be devastating and dramatic. This is Xanthorrhoea johnsonii, I think, up at Werrikimbe National Park, just inland from Port Macquarie. So very devastating in these very susceptible species. Ed Liu did some work on the Wollemi pine, and you can see it's an enormously good indicator species for the presence of Phytophthora in a potting mix, and the symptoms develop dramatically and aggressively and are quite devastating in that situation. And of course, it's problematic in terms of the cultivation. Ed's also done some work with Keith McDougall looking at the reaction of a number of threatened species to Phytophthora, and as you can see between the control plants on the right-hand side of the screen and the infected plants on the left-hand screen, the impact of these sorts of pathogens on many of these threatened species can be dramatic and huge. And just to highlight the problem, this is the impact of the disease on the Phytophthora, on the Wollemi population in situ, where the original populations were discovered, and unfortunately the pathogen was introduced into the soil a number of years ago, and is a problem in terms of managing that site. We've been doing some work recently looking at the impact of Phytophthora in urban forests and some of the major tree planting programs that have happened in Sydney. This is the sort of scenario we often see in this case with Phytophthora cactorum, and we've done some preliminary work with a relatively large number of samples now, and coming out at about 23.6% of nursery stock coming up Phytophthora positive, with about 5 different species. So it's a big problem, and probably a undetected problem at this point in terms of these urban forest plantings that needs to be addressed. And as you can see, I put this one in just to show the sampling process that we do for detecting Phytophthora in the potting mixes now, with these in the middle there, you can see these sort of modified trowel type situations. But the one point I wanted to make there was particularly the shot on the left-hand side is a really critical thing that needs to be avoided is storing pots ready to go back out into the ground, back out into the ecosystem straight on the ground. They need to be on elevated benches so that we can prevent the splash and the cross infection of plants being affected by Phytophthora. And this is just a quick shot of the detection system that we're using now. It's all involving quantitative PCR detection and phylogenetics. High level of accuracy now and high level of detectability, and we can, using this system, differentiate down into different species, so we have a particularly fast and rapid detection system. It is worth pointing out that new Phytophthora continue to be discovered and continue to be causing problems all around the world. My New Zealand colleagues don't need reminding with the devastation that's happening in New Zealand with the kauri, but sudden oak death on the west coast of the US, and now in Washington and California, Oregon states, a huge problem, and new diseases like Phytophthora pluvialis around the world, all problems. So there's a continued need to detect and prevent these infections happening and new incursions happening. So it's not a problem that's going to go away. It's not a problem that's going to solve itself, but it is one that needs to be considered, particularly in terms of making sure that you're growing plants cleanly, carefully, and very judicious in terms of what you use, particularly when you go to the process of going to a translocation, so that when you do plant new plants back into the wild ecosystems, that you're not also introducing pathogens like Phytophthora. And I think that's my end of my presentation. Thank you.
Thanks, Brett. Any questions? Just raise your hand, a little icon, if you have a question for Brett. No, maybe you're still mulling over some of that. Oh, Amelia. Go on, Amelia.
Yeah, so Brett, I was wondering, the nurseries that you said you've been surveying at the moment, what kind of nurseries were they? Are they commercial, restoration, council?
I think at this point I can say, without trying to drop anybody into it, a name and shame sort of scenario, it's been a combination of commercial nurseries, community type restoration nurseries, and particularly working with councils to make sure that their material that they're getting in and then planting out is clean. So working with them to get better holding, particularly in terms of holding situations.
Yeah, great. So quite widespread.
Yeah.
Thanks. Yeah.
All right. Don't think there's any other questions. Oh, Chloe?
Yeah, hi. I just had a question about the Wallamai pine study that you did. How old were the trees that you did the study on, and how long did it take for the Phytophthora to, you had the 4 images there of the plants, how long did it take for that to happen?
To be honest, though, I can't exactly remember the age of the plants. They were, they weren't they were still relatively young, but they were well established. But the frightening part was the speed with which the symptoms appear. It's enormously susceptible, and we're talking a matter of weeks to start seeing symptoms, and then only a couple of weeks to see symptoms, and then death of the plant was probably within 6 weeks at the max. Yeah.
Right. Yeah, that's really quick.
So they're super sensitive.
Yeah. And just one more quick one. In the other images you had of the trials, you did it look in the little cuttings on the threatened plants, the Prostanthera looked like it had totally been killed, but the Nematolepis looked like it was a little bit more resistant to it. Didn't look like it had been completely killed.
Yeah, that's a fair point. They do have a tendency to be quite variable in their response. So you'll see some species which will have really good tolerance and able to fight off the disease quite well, and some that are super sensitive. So in that case, the Prostanthera was pretty sensitive to it, but the Nepalipis was much more tolerant. And there'll be species that will be have much less damage on their roots than those.
Yeah. No, thanks. So it's such a hard disease to control, and prevention is probably the best type of control for it.
Absolutely.
Yeah, thank you.
Great questions. So thanks. Right, so now we're going to hand over to Bob. Amelia, am I showing these PowerPoints, or are you?
If you could do that, Emma, that would be great.
Right.
Thanks.
I will.
Thanks, Emma.
It's all right. 1 second.
Okay, my name is Bob Makinson. I'm a conservation botanist, and I should say straight up that I'm not a plant disease expert, although I have a certain amount of knowledge on a couple of them. But I have been involved in a whole bunch of conservation-related projects, including threatened species work, where disease has been a consideration. So I'm coming at this from that kind of point of view. Next slide, please. Slide. Thank you. I'm not quite sure of the cross-section of audience we've got today, but people are likely to be working in collections of various sizes and types, and the type of collection obviously relates very strongly to what your goals are and your end uses for your plants, whether it's display, conservation, ecological restoration, or whatever. And that's going to shape your considerations about disease planning and response. That will also be shaped by the kind of species you've got and their characteristics, the diseases that you think are likely to be a problem for those species in your location, and then considerations about which diseases are simply ambient in your area and you'll have to deal with them on an ongoing basis, which ones come from your supply chain and can be managed in that kind of arena, and which ones might depend on or be vulnerable to manipulation in your particular collections, location, housing, or management technique. Next slide, please. How much you can do, of course, depends very much on budget, staffing, and competing priorities. But within that ambit, what you can do is going to relate back to those characteristics we just looked at about physical size, layout, extent of the collections, and composition of the collections, the ambient environment, and the end-use goals that you have. But there are other things like neighbour, visitor, manager, meaning your line manager considerations. Those people have got to be on board very often. And the effects of disease prevention and treatment options that you may have available have to be taken into account. There may be end user stipulations on contracts that you've got for supply of material, and you may have a corporate or institutional philosophy. For example, some regional botanic gardens have a no chemical ethos. And that's obviously a major consideration. Next slide, please. As much as possible, it's about anticipating risks because, as Brett pointed out, prevention is better than cure. But that is going to fail from time to time. And when it fails, having a prepared response, or at least a prepared body of knowledge about a suite of diseases most likely to affect you, is going to count for quite a bit. And you need to factor disease risk into your plant acquisition strategy, your planting layout strategy, collection management techniques, and the end use considerations, which we will come back to later. Obviously, the more you can study likely or high impact diseases ahead of time, the better, because you can build up a bit of a knowledge base for those with recent reviews or papers, or better still, contact with experts. And it's worth considering whether you can designate somebody in-house to play that role, to become a knowledge broker, or at least a knowledge accumulator on a range of diseases that you consider as priorities. It's a good staff development exercise if you don't have a dedicated staffer already doing it, but it's also the sort of stuff that a skilled and sensible volunteer can do, depending on the size of your operation. Cultivating experts is really important, and reaching out to them before you have an emergency is a good thing. But then there's training and preparation. Do you know, if you have an outbreak of disease X, who's going to do what? Are you in a position to consider using chemicals? Is that a reasonable response to take? And if so, have you got the gear? Are people trained and on board about doing that? And more broadly, with your collection, because it's a valuable asset, what are your parameters for triage and salvage of that collection? If it's a threatened species, what are you going to do with the surviving material, and are you going to shift it to another location? Things like that. Next slide, please. So it really helps, certainly I've found it really helps from an end user point of view to try and break the process down into component parts and analyse the whole chain from acquiring plants through to end use. And the initial acquisition phase may come via collection from the wild or from cultivated sources. It may come through exchange or donation, or it may be purchase. And one of the first things to look at is avoidance of transport of disease to or from the collection or purchase point back to where you are going to put it and keep it. In the case of actual collection activities in the wild or in gardens or whatever, disinfection of equipment between sites, and often also between plants and between species, is important. And there may be particular precautions that have to be applied if you're going from a contaminated area like the east coast of Australia to a non-contaminated area like the west coast of Australia in the case of myrtle rust. When you're sending staff or volunteers out to do this kind of collecting work or acquisition work, don't just trust to luck or rely on people doing the right thing instinctively. It's worth auditing your processes and running them through it on a dry run, and make sure that they are following the protocols that you have adopted as appropriate for your gardens. If you're buying stock, be aware that all supply chains have to be considered somewhat suspect until proven otherwise. The problems exist across the whole spectrum of potential suppliers. And it's best to know your supplier and their systems, and asking to see their process, if it's within physical access, or their standard operating procedure documentation, is a good thing to do. It puts them on their toes, and it gives you a much better idea of where the risks are upstream of you. So auditing upwards along the supply chain is really important, and stipulating hygiene in contracts is a significant step that you can take, which gives you some leverage later on. Problem resolution may be awkward, but it is necessary. It may need senior management involvement. It may involve inserting yourself into a process which has been done on a handshake between two other people, and that can be quite awkward and sticky to do. But if you don't do it, you're not going to solve the problem, and it's going to continue to be an issue. If you are an end user provider, and that includes the display of plants to the public, as well as the supply of physical plants, audit down the supply chain. How is your stock viewed after end use, and how is your reputation as an aware phytosanitary institution affected by what's happened? Next slide, please. So continuing along the spectrum of plant acquisition and management, then we come to the reception stage, when the plant is carried in your front door. And ensuring sound hygiene practices at that point is very important, particularly if you have severe disease potential. The direct delivery of incoming plant material, that is not having it stop off along the way at various people's houses or nurseries or whatever, is very useful because it minimises time in transport when it can pick up ambient spores or be put on the ground and collect something that it shouldn't have. And if at all possible, there should be some level of quarantine when it arrives at its destination. Not everybody has a dedicated quarantine glasshouse or anything like that, but some level of quarantine and segregation from other plant material, particularly of the same species, is usually doable for a period of time. And during that time and afterwards, you need to monitor regularly. And it helps to develop a checklist of pest and disease symptoms that are relevant to the species that you have, and to train staff and volunteers in what to look for. Now, a lot's going to depend on space, resources and risks. I won't dwell on these because I'm going to run out of time, but it's on the slide there. Some points are on the slide for consideration later on. But it's worth noting that the development of a short list of infrastructure, equipment and training that you might think are desirable for phytosanitation is developed, and that you keep that in your bottom drawer or your top drawer indeed, with a view to funding opportunities that may come out of nowhere, like end of financial year underspends and things of that sort, because you may be able to acquire quite a lot of gear there that's not on your general budget. It's also important to consider broad spectrum low level disease, not just the big killers like Phytophthora and myrtle rust and things of that sort. Okay, next slide. This is a bit of a sidelight, but if you have what I'm calling here variability collections, there's other terms for it, but essentially collections of a genus or of a species that have been brought together for assessment of some variable character, let's say it's flower colour for ornamental purposes. Be aware that that collection, particularly if the provenance information is good, may have particular value for disease assessment and the detection of differential levels of resistance or susceptibility to disease. This lemon myrtle plantation in southeast Queensland was developed to test for the best essential oil genotypes back in the 90s, and it played that role. It had material from all known wild provenances of the species. But when myrtle rust arrived, that hit the lemon myrtle industry quite hard. And fortuitously, this plantation had survived, and most of the documentation associated with it had survived by a small miracle. And it acquired a whole new relevance as a chance to assess different genotypes against the myrtle rust pathogen. And some indeed proved to be more resistant than others, and it's going to be a factor in the recovery of that species down the track. It's worthwhile, if you have a collection like this, getting to know researchers with an interest in that species or genus, or with the diseases that affect it, and network with them and just let them know that you have it, because they may be able to devise a project that helps you sustain it financially, who knows. Next slide, please. A couple of other factors. Publicity around disease and pest questions can be problematic. The case at the left of the screen there was actually a pest, not a disease as such, back in the early 2000s in Sydney Domain, where a number of fig trees were getting hit by bad psyllid infestations year after year and were becoming unsafe. And the proposal, which was well aired and well consulted and a very transparent process with neighbours and stakeholders and the public at large, was to replace them with some other species. However, for various reasons which don't get me started, some parties went on the warpath about that, and there was quite a set to, and the management body for that site had to be, unfortunately was, well prepared to provide a strong argument for why it had to do what it had to do. The case on the right gets back to this problem of handling and negotiating with supply chains who may not have a lot of money to throw at phytosanitation, but may be a critical supply chain for you. I won't name the species, but this is a threatened species, and a not-for-profit nursery was supplying material for a translocation program of it. And they wound up, due to a shortage of bench space, of putting some of the mother plants that they were using for cutting-based propagation, on the ground. And they picked up this disease, monocytia, which is a raindrop spread fungal leaf fungus infection. And some of that made its way into the translocation stock and one of the translocation sites. At least that's our reconstruction of the process. It's plausible. I'm personally convinced that's what happened. But because everything was done on a bit of a handshake, it was hard to sort of read the riot act about benching plants in as strong a way as one might have liked. So contracts are good that way, and stipulating contracts what has to be done. Next slide, please. Just about finished now. Finding good information does take time, and don't take the first thing you read as gospel. We all know that. The two addresses that left there, and I apologise for the New South Wales centrism, but they illustrate the point. Major botanic gardens are one of the contact points for good information and likely current information. And the other one is agricultural agencies in New South Wales. It's local land services and the primary industries department. But NRM bodies, natural resource management bodies around Australia may have it. And of course, New Zealand has similar bodies as well. The industry and community sources are important, and it's good to reach out into those and have prior connections. The APVMA in the Australian context is the body that can talk to about off-label permits and things of that sort, but also talk to your primary industry agency. The Hort journals and the horticultural industry representation bodies are, again, worth having a network in, but be aware that the publications quite often are fairly out of date, certainly as far as details like rotation of best fungicides go and things of that sort. So the three publications shown there all have material relating to myrtle rust, but it all derives from around about 2012. And the fungicide and rotational scheme has moved on since then, and you will need to seek out what the most appropriate stuff is. Next slide, please. And that's my lot. I'd just like to say thank you to Veronica Weiler here at Mount Annan Botanic Gardens, where I am, and Darrawal Land, and Brett Summerall for their comments on the draft for this talk. Thank you.
Thanks, Bob. That was really interesting. I enjoyed hearing about the lemon myrtle example. It's a good reminder of some of the usefulness of collections we hold. The usefulness might not be apparent right now, but they will be at some stage. Does anyone have any questions for Bob at the moment? Just check the chat. If not, feel free to pop them in the chat and we might have time at the end. Otherwise, I'm going to hand over to Beck Stanley.
Thanks, Emma. Kia ora koutou. It's lovely to see you all again in this BGANZ forum, and great to have this opportunity to promote the germplasm or parts of the germplasm guidelines with you. They're a fabulous resource. I think they're both technical and practical, which is pretty hard to actually achieve in a document, and I think it has. So the work I'm about to talk about today is some of the case studies that Emma and I worked on at Auckland Botanic Gardens. So it kind of brings to life some of the things that Brett and Bob have already mentioned. So obviously establishing ex situ collections is a first step in plant conservation. And this, as I said, this talk discusses some examples of what Bob and Brett have already highlighted to us in this session. So receiving material as an ex situ provider of a plant that's highly susceptible to a pathogen can be a really complicated gift. The provider needs to ensure they can give proper care to that affected germplasm by working out a regime specific to that, and ensuring resourcing to do that, and assessing all the risks that might bring to that facility. And that includes talking to all the stakeholders who might be already involved with your organisation on other projects, and approval by managers and acceptance by staff of some of those different care regimes that might be required. And of course, we also have to think about protecting the existing plants, collections and projects at the facility. And I'll talk about this in a bit more detail, disposal options if you need to do that. And the facility you approach might also need to consider that they might not be the right provider to take on a threatened species. And especially if you've approached a BGANZ institution, that's a great way to start, because they might be able to broker a better or additional place for that collection. So my first case study is on Rata moiho Metrosideros bartlettii. So Auckland Botanic Gardens received 5 genotypes of Rata moiho of the 12, or sometimes people say 13 plants left in the wild. And it was sort of out of the blue with 4 hours notice. It related to a helicopter collection of material actually for DNA analysis. And then when they looked at the material, they thought, hey, some of this could be propped. So sort of phone call at 6 a.m. from the Conservation Authority saying, can we drop these off to you this morning? And of course, you know, this is the kind of situation where it's very difficult to say no, because you want to help, and you feel, you know, quite lucky to be given this opportunity. So at the time that was fine. However, a few years later, myrtle rust arrived in New Zealand, and you'll be able to notice immediately from that flower, this is Metrosideros bartlettii and myrtaceae. So suddenly we had a new approach was needed to be considered in terms of just having this thing and not really doing much, to having this thing and needing to do things quite differently. So first of all, we had to trial some suitable fungicides. This plant had never been sprayed with a commercial fungicide before. The first couple of trials ended up defoliating it. So that also shows the benefit of being able to do this stuff in an ex situ facility. We did end up finding fungicides that didn't defoliate the plant. Then we had to start this preventative fungicide regime. Now, Auckland Botanic Gardens prides itself on being relatively chemical free. In fact, we say we're pesticide and fungicide free. So that's quite a shift in practice. A lot of staff are very proud of not using chemicals, and it required, of course, training and certification and using them every 3 to 4 weeks. It also meant we started hand watering the plants compared with having automated overhead systems, and that was to reduce the conditions that were suitable for rust. So immediately hand watering, you already know what I'm talking about there. That's a person every day or every other day actually standing there watering the plants, which is a massive resource. We also house them separately in a shade house away from the main standout area. We felt like that would be another physical barrier to inoculum to keep the plants clean, and it kind of also stopped us moving from where they were to the main collection. So it potentially stopped any inadvertent movement of the rust on our clothing or our tools. And Emma also set out to duplicate the collection to a facility, which at least for now is outside of the current range of the rust. So that's some of the things that we had to do when we weren't planning to do any of that at all. Of course, we have then thought about, we're not just keeping these plants forever in an ex situ facility, we are wanting to return them to the wild. So this is a photo Emma took of some of the iwi from the location in the wild where this plant's found, up in the far north of New Zealand. So we need to ensure that we keep that material clean, and that is using fungicides and having a vigilant survey regime to detect infection, to be confident that clean material is returned to the wild. So this feels especially responsible because unless you've kept up that preventative fungicide regime, you don't know whether there's an infection you can't see. So having those records and that regular regime is really important to be able to give quality assurance that you're not taking these things back infected. So the lessons learnt were ensuring that we have the skills, tools and resources available to look after this threatened plant with a disease or likely to get it. That staff are qualified to use fungicides. As I said, it's a facility that usually doesn't use them. We needed this bespoke watering regime. And we had to weigh up the costs really of managing this plant in an ongoing sense, like potentially indefinitely, and thinking about the time that takes to do these extra treatments and the space that it needs to be separated from other collections. And then of course COVID, which, you know, none of us could have expected, but really you have to think about being prepared for the unexpected. Hand watering wasn't really possible when we were prevented legally from actually going to work. So that in the first COVID lockdown, that was my main stress, was that we had genotypes of plants that were nowhere else, that were required to be hand watered every other day, and an absolute lockdown where no one was allowed to go to work. So in the end we brokered it, but there was a very stressful week to try and make sure that we could get those plants cared for, and they were. But you know, these things are not as simple as they might seem. So my second example is prior to the arrival of myrtle rust in New Zealand, the gardens established a seed orchard of wild collector Lophomertus, which is a bit like your rainforest shrubs that are getting myrtle rust. So we learned from, they're called Rhodamnia and Rhodomyrtus, I think. So they in Australia were getting infected, and we thought actually the most similar thing we've got here is to Lophomyrtus, and we think potentially it will be also highly affected, and we were right. So prior to the arrival of myrtle rust, we thought we'll collect lots of material from the wild in Auckland and cultivars. This is a popular plant developed for cultivation, including natural hybrids, because we'll see which ones are infected and which aren't infected. Now, this is completely my fault. What a naive thing to think, because what happened was it was in the nursery, hadn't yet been planted out in its orchard when myrtle rust arrived in the country. And the seed orchard, as we expected, these plants were highly susceptible, and they were one of the first victims of myrtle rust at Auckland Botanic Gardens. We were members of the New Zealand Plant Pass scheme, in fact, some of the first members in New Zealand. And the nursery hygiene scheme really focuses on not harbouring disease hosts in your nursery. So this kind of having even the presence of this ran really contrary to this approach. So it seemed like a good idea at the time, but on reflection, we really hadn't thought it through, and we should have thought it through, because on that day, Emma and I were standing in the nursery going, what are we going to do? Are we going to chuck out all of these plants that are potentially nowhere else? You know, what should we do? What's the best thing with this rare genotype? So we quickly assessed that the infection would put other crops and collections at risk, including this. So this is the Auckland Botanic Gardens nursery grows 60,000 native plants every year for revegetation projects back in the wild, and the majority of them are Myrtaceae, Kunzea and Leptospermums, and Metrosideros.
Okay. Yep, that's fine.
So they were potentially all of that is going to get infected with myrtle rust. So the stakeholders for those plants, the people who were getting those plants, the rangers on parks, were unhappy that we would keep the Lophomyrtus orchard in a place where their plants were being grown. So we thought on our feet. We knew it was a valuable collection of genes, and we knew that some of the cultivars weren't infected, and we were sure that that was probably interesting to someone. We wanted to deaccession them, but they're valuable. So really quickly, we organised the transfer of these plants to an off-site research collection, which is managed by Rob Beresford, who is a plant pathologist undertaking research into myrtle rust resistance in New Zealand. So we found a win-win, really. We called him, we said we need to, after Emma and I, should I admit this, Emma? After Emma and I had stuffed them into rubbish sacks, we contacted Rob and said, oh, we were thinking of throwing these in the skip, but we think maybe you might need them. And he did want them, and he planted them out, and we've ended up, the three of us, writing a paper on susceptibility of these horticultural cultivars to myrtle rust based on his orchard, which is a long way from a revegetation collection at a research facility somewhere else in Auckland. So bear in mind, you have to have permission to be able to move that, and he did have permission to move it. So it was kind of a win-win, but I wish we'd thought of it first, because that morning was pretty stressful. So our lessons learned were really talk to the ex situ provider as early as possible if you're going to bring a plant susceptible to a disease to a facility. So they can talk to their staff, their managers and their stakeholders about implications of the acquisition, make a plan, and as Bob said, not a handshake, for how you will safely care for that collection, minimise the risk of infection and spread within your facility. Make sure you decide ahead of time what happens if the material gets infected, to avoid disposing genetic material which may be on the verge of extinction or really valuable to somebody else, like a researcher in an approved facility. And duplicate the collection to spread the risk if you can, and ensure that place knows all about how to care for it. Both of these case studies are written up in the Germplasm Guidelines, case study 11.8, and Emma and I have also written it up in the Australian Plant Conservation Journal. Thank you so much.
Thanks, Beck. That was great. Are there any questions for Beck about her case studies she's presented?
You might ask me.
Yeah, I could answer them too. No burning questions. Amelia, did you want to say anything?
I just wanted to say thank you to Brett and Bob and Beck for the presentations. That was fascinating. Hair raising, shocking, all the things that can be associated with plant disease. I remember as an undergrad pathologist like Brett and his peers got, that is just such fantastic plant disease, but it's devastating in conservation collections and in the wild. So we're really lucky that we have a chance to have questions. And with Bob at the Australian Plant Bank there, I think we also have Veronica Weiler, who's a horticultural research assistant, and Veronica works day to day with the plant collections there and does a lot of that hands-on work, making sure the collections are healthy and well maintained, so she can answer questions as well.
We have got one in the chat specifically for you, Brett. Is there much known to the physiological or mechanical reasons why certain taxa or populations are more susceptible to being killed by Phytophthora than others?
Yeah, that's a really good question, because there's yes there is and yes there isn't, I suppose, is the simple or the complicated answer from that. We don't really know the genetic basis of it, and it does seem to be, like many soil-borne diseases, controlled by a number of different genes and mechanical processes. Certainly when we apply chemicals like potassium phosphate, Phosgect or anti-rot, as people might know, we know that the enhanced defence responses that get stimulated in those sort of circumstances mimic in susceptible plants what you might see in resistant plants. So there is probably a whole range of different chemical, physical and mechanical mechanisms for the different levels of disease response and tolerance that you see in different species.
Great. Any other questions, Bob? Oh, you're just on mute still.
I think it's worth making the point in regard to that last question and answer that our capabilities in terms of figuring out what's going on, not just genetically but physiologically, are improving on an almost exponential curve. The metabolomics and various other techniques known collectively as the omics almost give you an ability to look at physiological processes, including disease, challenge and response, almost in real time. It's just phenomenal. And coupled with genetic and epigenetic insights, we're going very soon to have a vastly improved understanding of the workings of both disease and resistance in plants. It's going to take a while, it's going to be resource dependent, but the curve is upwards. Now, that doesn't necessarily translate to a high-tech solution, but it will give us a much better idea of what's going on, and it will facilitate other techniques like RNAi vaccines, which are a very interesting development, applied so far mainly in certain horticultural areas and of short duration. They're not a magic bullet for everything, but our capabilities are expanding very, very fast indeed. So while plant pathology is traditionally an arena of doom, gloom and despair, that still exists, but it coexists with some bright side as well.
Amelia?
Yeah, thanks, Emma. I actually have a question for Veronica. I know Brett alluded to these really, the flooding conditions and how, the potential impact of Phytophthora. But I was wondering, you manage collections that are susceptible to myrtle rust. How does the management of those kind of collections change in wet conditions like the La Nina that we've been seeing recently?
Thanks, Amelia. Well, significantly. So with this current wet summer, that's meant that we've had to be on a pretty steady rotation of fungicides for the collections, and that's pretty much been avoidable. So that's two years that we've actually had to do that now, and as these sorts of events become more common, I think it's going to become more and more challenging across all diseases, but also pests. So we've noticed a marked increase in the nursery of pest numbers and outbreaks. So yes, I think climate change is going to be a challenge across pest and disease management broadly.
Thanks, Veronica.
Awesome. Is there anything else from anyone they'd like to share? Questions, comments, queries? No? Okay. Amelia, do you want to wrap up from your side?
Yeah, fantastic. So thank you to all the presenters today, and thanks for everyone for joining us online. A big thank you to the Ian Potter Foundation, who provides funding for the Germplasm Guidelines revision and this webinar series as an extension of that, getting the information out there and, you know, adding to the information. I think in Bob's presentation, he pretty much scoped a new section of the next edition of the guidelines. Thanks, Bob. I'd like to invite everyone to join us at the upcoming conference of the Australian Network for Plant Conservation. APCC 13 will be held from the 3rd to the 7th of April in Albury and online, and we'll share the link for that. And a very big thank you to Emma for hosting today, because Emma has a really exciting event herself coming up on the weekend. She's about to go off and get married. So we wanted to wish her all the best for a very happy day and many happy years to come. Thanks, Emma.
Thanks everyone for coming to my last official thing for work before I am officially married. So just to let you know that the April Plant Forum will revert back to our previous format, where we will have 3 speakers. So we've got one from Eastwood Hill Arboretum and Wellington Botanic Garden in New Zealand, as well as someone from Geelong. So that's very exciting. And before you disappear, Rebecca Harcourt just wants to mention some of the upcoming Botanic Gardens Day events.
Hi everyone, I'm from BGANZ, and I just wanted to remind everybody about Botanic Gardens Day happening on the 29th of May. And the whole month of May we've got various events going on. I will just bring them up on my screen. So what is most relevant to you guys, on the 12th of May, Amelia and Emma are sorting out a webinar workshop day. I think it's 9:30 to 3:30. Correct me if I'm wrong, Amelia.
We'll just sort out the times, but yeah, right date, 12th of May.
Yeah, 12th of May, and yeah, Amelia and Emma are sorting out speakers for that day. It's going to be an all-day workshop for professionals. On the 5th of May, we have a webinar in the evening. Basically every Thursday, the 5th, the 12th, the 19th, the 26th. Keep your eyes open on the BGANZ newsletter. And we look forward to everybody participating in the Plant Challenge. The topic this year is Plant Passion, and do your little videos. We'll send out some info on that. And we're also going to have some prizes this year because we've got a sponsor. So that will be even more incentive for you to be involved. Thanks, everybody.
Thanks all for joining. We'll see you next time.
Thank you.
Thank you. Bye.
Thanks. Bye.